the first lightning talk of this afternoon session, the second session, is from Ana Paula Vargas Ruiz. Ana Paula will talk about comprehensive virtual screening of 4.8K flavonoids reveals novel insights into allosteric inhibition of SARS-CoV-2 MPRO. So, Anna, are you there?
Yes, can you hear me well?
Yes.
Awesome. Let me share my screen.
Yes.
Okay, can you see that now?
Yes, just, okay, now it's a presentation.
Okay, let me just, okay, great. So yeah, thank you, Wendy, for the presentation. My name's Ana Paula, I just usually go by Paula. And I'm a current graduate student at UC Berkeley, but today I'm gonna talk about some work that I did at the CIMIC lab last year in Peru at the Universidad Peruana Cayetano Heredia. And so, yeah, thank you very much for letting me share my science with you guys. Okay, so we already know, right? We're in the midst of a COVID-19 pandemic, and the virus that has caused it, SARS-CoV-2, is responsible for the deaths of more than 4.5 million people worldwide now. In Peru, the situation is it's getting better, but also almost 200 people have died. So yeah. So while vaccines have already been developed and are enabling us to combat the virus in the long term, effective and specific treatments have not been found. And this other goal will require like systematically selecting drug targets that can act against a particular element of the virus. And these targets can be like many different things, but one of the most common that has been proposed for SARS-CoV-2 is a major protease, or MPRO. So the main protease is the enzyme that performs the main catalytic activity of the virus. This protease will cleave the virus polyproteins here in blue, and that allows its functionalization. So in that sense, it's logical to think that if we manage to inhibit the MPRO, we would stop viral replication. And so this is unlikely to be toxic to humans because there are no human proteases with similar cleavage. And so a common technique to find inhibitors is to redesign those that inhibit similar proteins. And in this case, SARS-CoV-2 main protease maintains a high structural similarity with that of SARS-CoV-1. And it's actually identical except for 12 amino acids, and the 3 sites that comprehend its catalytic activity remain intact. So that is pretty important. So hopefully I have convinced you that the MPRO is a promising target for inhibition. And okay, so what should we use as drugs? And so in early 2020, there was a study that was published that evaluated the ability of 64 flavonoids to inhibit the SARS-CoV protease using FRET. So this was an in vitro study. And this study suggested that 3 of these flavonoids would serve as a basis for designing inhibitors. So flavonoids, as you may know, are these compounds that are secondary plant metabolites. So they present a variety of structure, like their chemical bases. I put them over here, but in general, they are characterized by being highly hydrophobic. They're polyphenolic. They have this heterocyclic ring. Anyway, so with this background, so with this study, and considering the high structural similarity between both proteases, that of SARS-CoV-1 and 2, we thought it convenient to evaluate all possible flavonoids as inhibitors. So we consider that flavonoids could bind the MPRO at up to 3 sites. So we consider the substrate binding site, which includes the catalytic diet here. It's a histidine and a cysteine. We also consider the dimerization site, which is at the interface of both monomers. And that is pretty important because the MPRO can only exert its activity as a dimer. So if we inhibit dimerization, we can also inhibit the activity. And finally, we consider a cryptic site, which is a site that can only activate upon the presence of a ligand. So these are kind of tricky to identify, but can now be predicted by computational methods. So we identified these 3 sites, we tested out flavonoid library against them. And you may be wondering why it is important to look beyond the substrate binding site. And there have been studies that show that there are some inhibitors that can bind that can impair MPRO activity binding away from the substrate binding site. So it's important to also account for them. Also, that dynamics of allosterisms are not really well characterized in vitro. So in silico studies like this could also shed some light on the mechanisms of action. Okay, so what do? We had this database that is composed of all flavonoids available in 4 repositories that were obtained by web scraping and data mining techniques. And we retrieved almost 7,000 flavonoids, which after being filtered by paints, we had like 5,000. And this paint filtering step is only to be sure that there are no super highly promiscuous ligands that could be false positives. So yeah, so with this almost 5,000 flavonoids, these were subjected to 2 rounds of virtual screening. The first one was a rapid virtual screening round, which we used Autodoc Vina for this, and we ranked them. So the top 1,000 flavonoids, we fed that to another algorithm, a second virtual screening, more exhaustive with Autodoc GPU, and that allowed us to obtain like the best 1,000 ligands per binding site. And our binding mode selection for each docking was performed by ranking them with a 2D score, which is a mathematical formula that allows to rank according to not only like the conformation of the pose of the ligand, but also accounts for the cluster population. So has 2 parameters in that. And so we accounted both for the binding energy and how often that binding energy occurred. So we chose then a top 5 ligands per site. So we had a total of 15 flavonoids that we were considering as candidates. And we then fed this into molecular dynamics study because docking studies can just show us, like we can only predict the preferred orientation of a ligand, right? But we wanted to also evaluate that through time. So we performed molecular dynamics and complemented that with MMB-PSA methods and SASA methods. And okay, so here's an example of the molecules with the best binding energy at each site, but this is only for docking. I'm going to show you molecular dynamics in a second. But this is just to show you, here's substrate binding site, this is dimer binding site, and this is cryptic binding site, and these are the best ligands that we found with docking. We had very favorable binding energies, -7 kcal per mole for the dimerization site, and for the other sites, a -9.5 kcal per mole. Interestingly, all of these substrate binding site ligands interacted with the catalytic histidine, and all of the dimerization ligands, all the ligands that docked against the dimerization site inserted at least one of their aromatic rings inside a cavity, a small pocket that was reported in other studies to bind allosteric inhibitions. So I'm gonna return to that in a second, but just keep that in mind for the next slides. Some of the cryptic site ligands also bound to this cavity, so we named that region A, but yeah, I'll get back to that in a second. Okay, so as I showed you in the last one, we selected like 5 candidates per site, so we have a total of 15, and then we selected the ones that remained bound during the majority of time of our molecular dynamics. I think I forgot to mention these are long molecular dynamics, so we performed 600 nanosecond dynamics on this. And so I'm just going to show you like the best candidates according to how many times they remain bound and the interactions they held. For the sake of time, I'm going to focus on some of this. So these are the 4 candidates that we proposed as promising: dorsolarin A, Ercanon A11, Sangenol O, and Campbell 98. These hold the highest residence time. As you can see here, this gray box just is the residence time. So at nanosecond 500, for example, this ligand Ercanon A11 would just dissociate from the protein, but the rest of them remain like for the whole dynamics. And what's important here, and I want you to note, is that the RMSDs seem pretty stable, like in general. Not only that of the flavonoid, which is in like the ligand, which is in blue here, but that of the protein and the complex itself. Remarkably, this Campbell 98 compound doesn't seem very stable at all. And I'm gonna also return to that in a second. And this is because this ligand kind of jumps through the protein and until it reaches its final conformation states at around 400 nanoseconds, and stays in another region that we also think it's allosteric, which is region B. But I'm gonna return to that. Okay, so yeah, we took snapshots of these. You can see here the green lines. We took snapshots of these molecular dynamics in order to compute the, to estimate the binding energies via MMBPSA, and I'm gonna show you that just to reassure that the binding energies were pretty favorable. So you can see here, this is dorsolarin E, Ercanon A11, Sangenol O, and Campbell 98. And this Campbell 98 had actually the lowest mean binding energy at minus 19 kcal per mole, followed by dorsolarin, for example. So yeah, you can see this, the binding energy is more or less the similar, while the dynamics just like go on, right? So, okay, so that is it by then. So I hope I have told you by now that these are promising candidates to keep exploring and maybe test in vitro, but because we did such long molecular dynamics, we can also explore how these impact the protein. And so I'm going to show you some of these results. So a couple of slides before, I said that all of the dimerization site, all of the ligands that interacted with the dimerization site inserted at least one aromatic ring, right, into a small pocket that I was going to call region A. So this pocket is reported in previous studies to bind allosteric inhibitors. And Sangenol O, one of our candidates, also binds to region A, which you can hopefully see here through the dynamics in this snapshots. And this caused constant fluctuations in the protein RSMD, which you can see here at the bottom left. This is just the same image I showed you previously. So these are constant fluctuations. As you can see here, the protein is in red. They're small, but they're constant. And this may be happening, we thought this may be happening because the region A is constantly being modified. And you can see here with the snapshots, right? Sangenol O like inserts its aromatic ring, and the protein just kind of wraps around it and opens up a bit the cavity until it has reached its final destination. So interestingly, also this ligand increased the RMSD of not only like the protein, but the SBS residues, so substrate binding site residues, those residues that exert the catalytic activity. And this is the RMSD that you can find here in red. So as you can see, there is a disruption of these residues at around 400 nanoseconds. This may be important because this region A is not very close to the substrate binding site. So this could be an interesting allosteric region to explore. And the last thing I want to show you, because before I run out of time, is what happens with this other ligand that I talked about, the Campbell 98, which promotes reorientation of the mains 2 and 3 of the protease respect to the main 3. So the mains 1 and 2, sorry, are the ones that are up here in this graph, and the main 3 is the one that I at the bottom. So in dynamics of this compound, which was originally docked at the cryptic site, we observed like an increase in the RMSD at nanosecond to 150-ish, more or less. And that would correspond to the reorientation of these domains. And so during this process, as you can see in this picture, the Campbell 98, we propose a mechanism by which Campbell 98 will contact this arginine 4 and will induce its rotation so that two polar contacts were forming between domains 1 and 2 and 3. And this induced rotation will orient the side chains of these two arginines, so arginine 4 and 2098. They will induce its rotation to a zinc conformation. So one in groups wouldn't be parallel to each other as we're here, but like more in a like in the same plane. And so the one in groups, yeah, being disposed like this would disrupt the interdomain polar contacts. And by this time, the compound would have already adopted its final position with a terminal like aromatic ring inserted between these two orange helices, which is region B.
Anna,
yeah,
sorry, Anna,
yes, it's
we
my
are on
last slide.
time, just to tell you, okay?
Yeah, no, I got a timer. I just, this is the last slide, but thank you. So yeah, just very quickly, this arginine 4 in the end will insert its side chain into this cleft, which would displace the other arginine again and impede the polar contacts to form again between the three domains because they are now disrupted. Okay, and this is, as I said, this is my last slide. So I just thank you very much to the organization. Thanks to my co-authors, Gabriel and Nicolas, the lab, and Counciltech for founding. And yeah, thank you. This research has recently been published, so you can Google it. You can write an email to me or contact me my Twitter if you have any questions. But yeah, I'll be happy to answer anything. Sorry about the 1 minute-ish time.
No problem. Thanks a lot for your talk, Hannah. So we have one question. So which program do you use to perform docking and molecular dynamics?
So docking, we used Autodock, and for molecular dynamics, we use NAMD.
Okay, thanks a lot. So I think that we don't have much time, so we will go to the second speaker in this session. Is Natalia Rivero from Colombia. Natalia will talk about the effect of CERN-P and CERN-AP composition on the structural properties of a model of the lipid matrix of the stratum corneum of the human epidermis. So, Natalia, are you there?
Hello, I am. Can you thank you for the opportunity? I share my screen.
Yes.
Can you see my screen?
Yes.
Thank you for your confirmation. Hello, everyone. My name is Natalia Rivero. I am from Colombia. Thank you for your participation in this conference. For a start, it's very important to explain you what is the stratum corneum. Remember that the skin is a stratified tissue. This means that the skin has layers. These layers are hypodermis, dermis, and epidermis. The epidermis has more 4 sublayers, and the stratum corneum is the most superficial. The stratum corneum are composed for tip cells called corneocytes, and these corneocytes are in a extracellular lipid matrix. These extracellular lipid matrices are composed for cholesterol, free fatty acids, and ceramides. These lipids are organized in villages, and these villages forms a repetitive pattern because each villager is in the top of the other villager. This repetitive pattern is very, very compact. For that reason, the lipid extracellular matrix prevents the loss of the water for dehydration, and protects the body against viruses, microorganisms, and UV radiation. But what is the specific reason for study this composition? For answer this question, it's important to remember that the ceramides are sphingolipids. These sphingolipids has two chains, a fatty acid chain linking to amino bond with a sphingosine chain. It's possible to combine 3 types of fatty acids with 4 types of sphingosines. In this specific study, use 2 ceramides, CERMP and CERAP. These ceramides have the same phytosphingosine chain, but the only difference is a group hydroxide more in the fatty acid chain in the CERAP. The specific reason for study this ceramide is because in skin disease, such as psoriasis, atopic dermatitis, and lamellar ichthyosis, the composition of ceramide is altered with respect to the normal skin. In this table, you can see this disease with the alteration in the lipid composition, and the common characteristic is that in the 3 diseases, the content of CERAP increased and the content of CERMP decreased. But in the moment, don't have any information about what is the effect in these change of the composition in the structural properties in the stratum corneum. And this information is very important for improve or develop more efficient therapies for treatment for this disease in patients with lamellar ichthyosis, psoriasis, or atopic dermatitis. In the 2018, Smith and college studied this composition with 2 models, with the composition CERAP 2:1 and 1:2. The authors only report one difference. This difference was the overlapping in the content with more CERAP. In this case, the authors report that the same thickness, and the author assumes that this minor overlapping is due to inclination conformation in the acid chain. But this result has a lot of limitations. One is the resolution in the network diffraction, and all the result was calculated only in 1 dimension. For that reason, our work developed a methodology in molecular dynamic simulation with the purpose analyze this effect in this composition. For this, the fixed step was obtain the models, in this case 2 models, with the same composition in the experimental study. This model has 3 villagers because this is the simplest model to ensure 1 villager in the middle of 2 villager mode. This is a repeated pattern in this stratum corneum. Each model includes linolenic acid and cholesterol and was hydrated with water in the external superficie and the middle of the villager. Next was developed the molecular dynamic simulation, field minimization, net equilibration, and final production simulation with 500 nanoseconds. This is one picture of one of the models. You can see that the full hydration and the lower hydration in the middle of the villager. And it's important in this point explain you what is the random walk. The random walk is a protocol improved to more in 2018. This protocol has an objective. This objective is improve the sampling and reduce the effect in the initial structural condition of the final results. In this case, the temperature change in a random space from Tmin to Tmax. In this case, in the first nanosecond, change randomly, and the last 1 nanosecond is reduced linearly to the Tmin. Next to obtain the trajectories, was calculate 2 categories of results, general structural properties and ideological bonding. The first parameter was the thickness. This is an important measure because allow us it's possible to compare the old result with the experimental result and all follow us is determine the model represents a villager in the stratum corneum in the human epidermis. You can see that age data for age villager in age model are in the range report in the experimental study. And in age values, you can see that the effect in the change in the composition, because in the model with more series P, the thickness is less with respect to the other model. You can see that this effect in age villager, but it's important to say that all the results was calculated only in the middle villager. In the middle villages, you can see that the thickness is less in the model with more CRP. This is possible because it's possible overlapping in the middle of the villager. To check that, we calculate the lipid mass density profiles. You can see that these profiles for each model, and you can see that the difference. In this case, you can see that 3 peaks. This means that are overlapping in the middle of the villager, specifically in the point 0. That point represents the middle of the villager. And this behavior is opposite in the other model. For that reason, this checks that the middle thickness is due to the overlapping in the middle of the villager. Another important parameter is the angle of inclination, because in the experimental study, the authors assume that the change in the overlapping is due to an angular inclination or inclinate conformation. In this case, you can see that for each ceramides in each model, the angle changed, but the change is different. In this case, the model with more therapy presents an angle near to 10 degrees, but in the other model, the angle is 4 degrees. In this case, it's possible to compare the experimental data, but it's different the analysis, because in this case, no is a straight conformation, it's a mirror inclination, but it's an inclination in both the acid chain in both ZAP and ZNP. For finish, in this case, remember that the only difference is a hydroxyl group more in the ZAP. For that reason, we calculate the number of hydrogen bonding formed per cell molecule. In this case, you can see that the change in the composition doesn't affect the number of hydrogen formed in this case. The number is the same in each model. For that reason, this change doesn't affect this behavior. Finally, the important conclusion in this study is in the model with bigger contains ZLP, presents lower thickness, greater overlapping, and more inclined acid chain. And it's observed the hydrogen bonding format doesn't change with the change in the composition, specifically ZLP composition. Thank you for the attention. I'm very pleased with the opportunity to talk in this wonderful event.
Thanks a lot, Natalia. It's time for questions. Oh. Is there some questions or? Okay, I will perform one question. So experimentally, how it's calculated the thickness, and you mentioned the reported thickness of the membrane, but I was not clear which layer did they measure experimentally.
Okay, in the experimental study has a lot of villages, near to 1,000 villages, and for that reason, the author reports the prometheus in all the villages. For that reason, in the simulation, we use a 3-villager model, with the intention has a more representative model with characteristics in the lipid extracellular matrix in the stratum corneum. because a model more glider is most difficult. The computational cost is more with respect to other minimal models.
Okay, thank you. Sergio has a question.
Hi, Natalia. Can you hear me?
Yes.
Oh, hi. So thank you very much for the talk. I just noticed that you have very little amount of water in the middle layers. This is, I mean, perhaps it's just my ignorance, but I find surprising that you can still maintain the right orientation with such a little amount of water molecules. I mean, how the hydrophobicity is maintained.
Yes, in this case, we put these water molecules in the middle of the villages because in the experimental study reports humidity, because the molecules in the atmosphere or environment are in the model, in the artificial model or experimental model. For that reason, for have the same characteristics, include these molecules in the middle of the villages. But the molecules have a different behavior in the external superficie with respect to the middle of the villages, because in the middle of the villages are in aggregation, in groups, and in the external superficie, the behavior is very different.
Okay, thank you.
Okay, thanks a lot, Natalia, for your talk. And the last talk of this session of biomolecular simulations, structural bioinformatics and system biology is from Janin Riedelsberger from Universidad de Talca in Chile, and she will talk about shedding light on the structural function of rice sodium potassium transporter in Oryza sativa HK2.2.
Thank you, Wendy. Just give me a second to share my screen.
Okay.
Okay, so can you see the presentation?
Yes.
Okay, great. So thank you for the introduction, Wendy, and for the organizers for letting me present my work. So I hope you all have some energy left for this last talk. I think it was a long day of very interesting talks. So in this last talk, I will present you a study that we have done on HKT22, which is a sodium potassium transporter from rice. And in the study, we combined computational and experimental methods to analyze the structure and how it's related to the function of HKT22. So in the beginning, I will give you some information about the channel itself. So HKT stands for high affinity potassium transporter. And this name is a little bit confusing nowadays because the first member that was characterized was actually characterized as a potassium transporter. But with the time when more members have been characterized, it has actually been shown that HKT channels are more often or more frequently transporting sodium ions, and only some of them are transporting sodium and potassium ions. So, but still the nomenclature sticks. So when I say that these proteins are transporting sodium, they are involved in or they have a role in salt tolerance. So if you see here on the right, there are some rice plants that have been grown over the course of 15 days at high sodium concentrations, 150 millimeter of sodium chloride. And you see most of them are dying actually, and they're not able to grow under these conditions. But there are a few of them that actually are able to grow. And so these varieties are actually salt tolerant, and in these varieties, very often HKT transporters are expressed. So that's why we are interested in studying the structure and the function of these proteins to be able to understand how the salt tolerance mechanisms work. So HKT proteins actually have a very simple, or a rather simple structure. They have 2 transmembrane domains and the pore domain in between them. And of these units, there are actually 4 in series, and they form one subunit. And this subunit is actually the HKT channel. And the members that have been characterized of this family, they are classified into different groups or classes. And this classification is based on the selectivity filter configuration generally. So here in this pore loop, there are glycines generally, and these 4 pore loops gather in the center of the protein, and they form the selectivity filter. And in the class 1 HKT channels, this first position, or this first pore loop, generally contains a serine. And these transporters or channels transport mainly or only sodium ions, while the class 2 HKTs, they have generally a glycine in this position, and they are able to transport sodium and potassium ions. So when we analyze ion channels in the lab, we use electrophysiology, and especially the 2-electrode voltage clamp, or TVC technique. This is a technique where we use oocytes from the frog Xenopus levis to express our channels. And then we use 2 electrodes or we pinch these 2 electrodes in the oocytes, and this setup allows us to clamp and to set or define the membrane voltage. And so we can measure the currents that are flowing across the membrane, which allows us to analyze the function or the motor function of our channels that are expressed in the membrane. So here are some example traces that we measured by characterizing HKT22, the Y-type channel, in the oocytes. And we generally test several solutions that contain different ion compositions to see how the channels behave under these conditions. So in these cases, we were varying the sodium and potassium concentrations, and we could measure the current responses to our voltage protocols. And so these are the responses to one experiment, to measuring one cell. And if we want to compare several measurements and do some statistics, we can actually plot the currents that we were measuring in response to the applied voltages in an IV curve plot. And this is what you can see here. So to characterize the HKT channels, we could actually identify 3 characteristics. So when we increase the sodium concentration from 3 millimolar to 30 millimolar, for example, we see an increase in the currents. In the IV curve plot, you can see that here from the white triangles to the white circles. So there's an increase in currents when we increase the sodium concentration in the medium. And when we add a little bit of potassium, we can actually, just 1 millimolar, we can actually see that the currents are doubling. So this is a difference here from the circles to the black squares. So this is a second characteristic that we could identify for our HKT22. And interestingly, if we further increase the potassium concentration to 30 millimolar, this positive effect of enhancing currents gets lost. So the currents that we measure in the presence of 30 millimolar sodium and 30 millimolar potassium is almost the same as the currents that we measure in the absence of potassium. So these are like the 3 characteristics that we defined for the Y-type, and this will be something that I will come back to later when I talk about some mutants that we are analyzing. So for the moment, it's important to understand that these HKT that we are analyzing is a class 2 HKT, because it can transport sodium and potassium, and the transport, interestingly, depends on the composition of the external ion concentrations. So now we are interested in understanding a little better the structure of this channel. So we were building some homology models based on bacterial KTR and TRK channels. These are also sodium and potassium channels, and they're very well characterized, and there are quite some structures, experimental structures available. So what we did is we did the homology model. We embedded in a membrane, and we built a system adding water, several ions like the cation ions, sodium and potassium, and the chloride ions. And then we performed molecular dynamics simulations. And here we performed 3 replicates of each 100 nanoseconds. So in total, we have simulations of 300 nanoseconds that we were analyzing for this study. And while analyzing the simulations, we were concentrating on the extracellular protein surface because we were interested in seeing how or where the ions would approach the protein before they actually enter the pore. So this is the top view of the protein, the pore in the center of the protein. And in the colors, you see some spots where ions were frequently approaching the protein. So there are quite some residues that are close to the pore entrance. Some are more in the periphery. And actually this was the spot that we picked to start a more detailed analysis because here was an amino acid situated, a lysine, that has been mentioned before in a study to be crucial for the function of the channel. But in this study, it was not possible to assign a biological function. So that was actually a very interesting spot for us to start with. So this is a very similar figure. In the top here, you see just for orientation, the pore. And this is the spot where 4 amino acids are located that we have seen that were frequently approached by ions. This is actually in the side view, you see it's quite far from the pore entrance, about 20 angstrom. And just in more detail, you see this coordination site is formed by a proline, 2 aspartates, and a lysine. And then we checked for the level of conservation for these 4 residues in the 8 HKT members that have been identified on Ruiz. We see that they are mainly conserved, at least these 3. And in the case of position 501, here is the negative charge mainly conserved in 7 of 8 HKT channels. So let's have a closer look on what's happening when the ions are approaching these coordination sites. So we see that sodium as well as potassium ions approach the site and that they are coordinated by oxygen ions. So we were calculating the distance between the ions and oxygen ions as well for comparison the nitrogen of the lysine. And the plots that you see here on the bottom are actually the example from when the sodium ion was inside the coordination site, and you see that the backbone oxygen of the proline is in close proximity to the sodium ion. In the case of the aspartate 75, there's always only one of these side chain oxygens close to the ion. So while one of the oxygens is close to the sodium ion, the other one is far, and from time to time it changes. So when the other one approaches, the first oxygen gets far, and then they change from time to time. While when you see the situation for the second aspartate, you see that actually most of the time both of the side chain oxygens are close to the sodium ion and are coordinating it. And as expected, the nitrogen atom of the lysine is actually never very close to the sodium ion. It's never closer than 3.5 or 4 angstrom, which was expected because it's a positive charge and they repel each other. This was just rather for the proof of concept, right? So from this analysis, we could identify that the oxygen atoms of the proline and both aspartates are coordinating the cations, and that the lysine is not involved in ion coordination. So what's the role of the lysine then? We found that the lysine is able to form salt bridges with the neighboring aspartates. And here, if we also calculated the distances between oxygen and nitrogen atoms, and throughout the whole simulation, both oxygen atoms of aspartate 75 are actually close to the lysine, And in the case of the second aspartate, there's always only one of the side chain oxygens close to the or forming a salt bridge, while the other one is far away or oriented towards the other direction and is not involved in salt bridge formation. But from time to time, the side chain is actually flipping. So this is the result for 100 nanosecond simulation. And when we average, or when we calculate the average in all three simulations, we see the same tendency, that the salt bridges are constantly formed. So now we characterize the coordination site, which ions or which amino acids are coordinating, and what's the potential role of the lysine, which might be to maintain the geometry of the coordination site by forming the salt bridges. So now we went back to the lab and actually generated some mutants. So we were mutating the 4 residues. And now we are coming back to the 3 characteristics that I was mentioning in the beginning. And we were actually comparing or analyzing whether these were affected or not in the mutants. So first, the proline mutant. Here we didn't see any effect. So this mutant behaved exactly like the wild type. And this was expected because the oxygen atom that is involved in the coordination is actually a backbone atom. So this is something that we don't change with mutating the residue. So this was totally expected. Now, when we mutate both aspartates by either removing the side chain completely or removing the charge, we see that there are differences in the functionality towards the wild type, but they're still pretty comparable. So we can see all 3 features in the mutants as well. So they are only sometimes a little weaker expressed. That was a bit surprising because we thought that if the ion coordination is affected, or if one residue is missing, that we would see a stronger effect. But probably because we did only single mutations, the second arginine could still fulfill the job. So actually, the most interestingly were the lysine mutants. Here, when we removed completely the side chain, or when we inverted the charge, we didn't see any currents. So the mutants were actually non-functional. So you can see here in the current traces, we couldn't get any currents. This is a comparison with the wild type. And 2 mutants. One is the arginine mutant, where we maintained the positive charge, but the side chain size was a little bit bigger. Or the mutant where we neutralized the charge. These mutants were functional, but they were quite different from the wild type. So let's have a closer look on this one. So here on the top, this is actually the plot that I was showing you in the beginning from the wild type. And here below, you see the experimental results from for the mutants. So the first characterization was that the currents are increasing with increasing sodium concentration. So in the wild type, you remember there was a significant increase, so about 5-fold. And if you check the mutants, here actually we see like a slight increase in the currents, but it's very, very low. So it seems that the ability of these mutants to transport sodium is actually affected. So let's see what's happening if we add a little bit of potassium. So on the wild type, there was a huge increase as well. So the currents were almost doubled. This actually, in the mutants, this feature is still there. And it's actually more pronounced than in the wild type. And when we further increase the potassium concentration, what was actually inversing the effect of this current enhance in the wild type. So we also see in the mutants that the currents are increased compared to the presence of low potassium concentration, but it's not as strong or not completely as in the wild type. So what we learn from this that these mutants of the lysine, they have reduced or affected sodium transport, while the potassium transport does not seem to be affected. So to summarize the this study a bit, we were identifying and characterizing this ion coordination site, where we have the two aspartates and the proline coordinating the ions. And the lysine was actually able to form salt bridges with the neighboring salt bridges. It might be important to maintain the geometry of the coordination site. And we also think that this lysine might be important to give a kind of impulse for the cations to move forward or to move the ions towards the pore. So what do I mean by this? I haven't talked about this in great detail during the talk, but maybe you noticed that I was talking generally about the cation coordination site and not about the binding site. So here, when we talk about HKT22, we are talking about an ion channel, and the role of an ion channel is actually transporting ions and not binding ions. So when we were analyzing the electrostatic potential of the channel, so here in the top view, you have the pore in the center of the channel, and the binding or the coordination site that we were characterizing here at almost at the edge of the protein. And when you see the red areas are like the negatively charged areas of the protein surface, and you see they are kind of almost like negatively charged paths guiding to or leading to the pore entrance. And the idea is that for the ion channel, it's hindering if the ion is tightly bound somewhere here. What is important for the ion channel is that the ion finds the entrance of the pore, so it can be transported across the membrane. So but our impression is what's the importance of this coordination site is basically the attraction of the cations, which might be realized by the negative charge of the aspartates. But it's not the objective to tightly bound it here at this or here at this position, but rather to push it along the way, along these negatively charged path towards the center of the pore. So in this scenario, the positive charge of the lysine might be positive or beneficial to do just this, to avoid that the ion is tightly bound here at the negative charges, and it can be like pushed along or towards the entrance of the pore. And actually, this is not the only negative charge or potentially negative charge paths, but there are several more that potentially might have a similar effect. So this is actually something that we are planning to analyze in the next step of the project. And with this, I will end the talk. Just thank the people involved in the work, which are Wendy Gonzalez, Ingo Dreyer, and Ariela Vergara from the Center of Bioinformatics Simulation and Modeling in Talca, Chile. And most of the experimental data were obtained in the lab from Miguel Pineros, who is working at Cornell University in Ithaca, USA, and the projects were funded by Fondesit and CONICET projects. So thank you for your attention, and I'm happy to take questions.
Thanks a lot, Janin. So if there's any question, Sergio?
Hi, Janin. Nice to see you.
Nice.
Nice presentation too. Question, I mean, is there any possibility that there is something about the dissolution of the ions because of the difference between sodium and potassium, so that this kind of pre-binding in a place which is not really the place where the ions will go into the channel would somehow facilitate the dissolution of sodium or something like that?
I haven't checked the dissolution, to be honest, but I'm not sure whether it's on the surface this is already an issue, or whether this might be an issue like closer to the pore entrance. So will be an interesting point to check. I will put it on a list. Thanks for the comment.
Oh, you're welcome. Thank you.
Is there any other question?
Okay, then I would like to thank Janin and all the girls that were today in the afternoon. And